So for this chapter, we're going to be talking about cells, and cells are the building blocks of the body. Cells have very different functions, so the function of liver cells are totally different than the function, say, of the thyroid cells. And that's why these different cells are known as differentiated cells, where each cell looks very different in order to perform its unique function. Here are just a couple of examples of these differentiated cells. So you can see here on the top, this is what a nerve cell looks like. This is what skin cell looks like, which are simple squamous epithelium, which they are just one layer of flat cells. And this is what a cardiac muscle fiber looks like. So you see, although these are all cells, they all look very different in order to accommodate their function. So again, that relationship between structure and function, and how the structure varies according to the function of the organ. In the next picture, you'll see a composite cell, meaning that this is not a real cell. But cells in general have three major parts. There is the nucleus that includes the DNA and controls the cell. There is the cytoplasm, and the cytoplasm is made out of a gelatinous material known as cytosol. And in that cytosol, you have all of the organelles swimming in them. And then there's a cell membrane that keeps the cell intact, which is the wall surrounding the cell. Okay, so we'll take a look at this composite cell. Here again is the blue nucleus with the DNA, the genetic material. You have, this is your cytoplasm with the cytosol, which again is that a little bit gelatinous material with all of the organelles that are swimming in them. And we are going to introduce these organelles throughout the chapter. And then there's a cell membrane on the outside that protects the inside of the cell and separates it from the outside environment. So the cell membrane, that's your outer boundary on the cell. It maintains the integrity of the cell, and it is known as selectively permeable cell, meaning that although it is a boundary, but it does allow some things to get in and other things to get out of the cell. And that's what we mean by selectively permeable, that it is not fully permeable. It does not allow anything to go in or out, but it selects things according maybe to the size of the molecule, according to the needs of the cells, and so on. It is also responsible for signal transduction, which means to receive and send signals. In order to do that, that would occur through the cell membrane as well. The cell membrane is made out of two layers of phospholipids. So it's a bi-layer of phospholipid. It does have some carbs, and there are definitely some proteins in there as well. So the framework is basically a phospholipid bilayer, where we have the hydrophilic heads, which are the water-soluble heads. Those are towards the surfaces. And in the middle, you'll find the hydrophobic tails. Being that there's lots of fat, the phospholipid bilayer makes that cell membrane permeable to lipid-soluble substances, so that anything that can dissolve in fat will be able to pass through that cell membrane. But if things are water-soluble, they will not be able to pass through the cell membrane. You also have some cholesterol that stabilizes the cell membrane. It kind of works like a skeleton for it. These cholesterol molecules are movable within the fluid mosaic of the cell membrane. So take a look actually here. So this is your cell membrane. We have the bilayer of phospholipids. This is one layer, and this is the other layer on top. This is the outside of the cell, while down here is the inside of the cell. So if you take a closer look of the cell membrane, here again are your hydrophilic heads towards the water that's found on the outside of the cell, and here as well towards the water inside. And then in the middle of the cell membrane, this is where you have your hydrophobic tails hiding, where there is no water. You can find some of these cholesterol molecules scattered throughout the cell membrane, and they can move throughout the cell membrane. So they are not permanently, their positions are not permanent. They can move from one place to another. These purple things, these are proteins. And you can find here that some of the proteins act like channels or canals or tunnels that allow for passage of things in or out of the cell. Some of these proteins are going to be enzymes. Some are receptors, which means that it receives information from the outside environment. Besides the proteins, we also have these carbohydrates. These carbohydrates are there for cell recognition and self-markers. So you'll find some of these, like these green ones, those are your self-markers, for example. And a self-marker means, excuse me, a self-marker means that that is the thumbprint of the cell. That's how the cell, the body, or your immune system knows that this is your cell, not to attack it, and that it is not a foreign body. Had this carb been a foreign body, your immune system will attack it. And that is really the whole basis of autoimmune diseases. In an autoimmune disease, the immune system is unable to detect these carb self-markers, and it would attack, the immune system would attack your own cells. Going back to these channels, any defect, there are certain diseases that are caused by the genetic defects of these channels. So for example, a mutation in the sodium channels, we'll find out that sodium ions are very important for the function of the nervous system. So a mutation in those channels can lead to the inability to feel pain, or sometimes extreme pain conditions. Mutations in potassium channels, well, potassium, again, we'll find out later on, is very important for the function of the heart. So any mutation in potassium channels can lead to the disruption of the electrical activity of the heart, which can lead to arrhythmias. It could also lead to impaired hearing. Abnormal chloride channels, that is the whole basis of the disease known as cystic fibrosis, where the mucus glands make a very, very thick mucus, leading to the plugging of different ducts. For example, it'll plug the bronchi of the lungs, leading to difficulty in breathing. It could lead to the plugging of the ducts of the pancreas. And it can also lead to a very salty sweat. Now the cytoplasm, and that's found between the nucleus and the cell membrane. As we said, it contains the cytosol, which is the fluidy kind of proportion part of the cytoplasm, and then the organelles, which are the tiny, like the mitochondria, the Golgi apparatus, and so on, that swim within that cytosol. The cytoplasm also has cytoskeleton, and that cytoskeleton is a framework of different proteins, rods and tubules that keep all of these organelles in position, and it also keeps the cell membrane, the cell intact. So if you go back to this composite cell, you can see that cytoskeleton. So all these rods, these little purple rods in here, these keep the cell intact and prevent the cell from collapsing. Those are made out of protein rods or protein tubules, and that is known as the cytoskeleton or the skeleton of the cell. So moving on to the cytoplasmic organelles. For each of these organelles, you want to know the structure. Okay, so what does it look like, and what is the function of each organelle? The first organelle to talk about is the ribosome. Ribosomes are made out of protein and RNA. They can either be freely mobile within the cytoplasm, so they're swimming freely in the cytosol, and those are known as your free ribosomes, or they could be on top of another organelle known as the endoplasmic reticulum, which we'll talk about in a minute. If they are on top of the ER, those that would make this endoplasmic reticulum, into what is known as a rough ER, because you could see that it has all of these ribosomes right on top of it, studded on top of it, giving it a rough surface. The ribosomes are very important for protein synthesis. Going on to the endoplasmic reticulum, these are tunnels, are membranes that make these all of these tunnels. It looks kind of like a maze. It's a very long tubular system. And within that tubular system, we are going to synthesize something. So the rough ER is important for protein synthesis, while the smooth ER is important for lipid synthesis. The smooth ER has other functions as well. So it also we'll find that it stores calcium, and this is a very important function in the muscular system. It also is important for detoxifying of different drugs. So we'll see that the liver, that is an important organ in drug detoxification, we'll see that liver cells have lots of smooth ERs. Now, the Golgi apparatus, that is also a sac of flattened membranes, and you want to think of the Golgi apparatus kind of like the UPS store of the cell. So protein will be made, say, in the rough ER, and then it goes into the Golgi apparatus. So in order to change its conformation, so remember we talked about the primary, the secondary, and the tertiary structures of proteins. All of these structures are, the primary structure is going to be made here in the rough ER. Secondary and tertiary, all that packaging is going to happen in the Golgi apparatus. And then the Golgi apparatus is going to literally give that protein a part of its membrane in order to cover and protect the protein. And now this is going to be released into the cytoplasm as a vesicle. So vesicles are sacs, membranous sacs, and the membrane a lot of the times would be actually coming from the Golgi apparatus. And these vesicles are going to store different things. They could store proteins, like in the case that we just talked about, but it could also store maybe a lipid. It might store, that protein might be a hormone. It could be an enzyme. So there are different things that the cells would want to store. So we'll take an example of an interaction between all of these different organelles. And in this, we're going to talk about milk secretion. So the manual or the instructions on how to make milk would all be here in the DNA of the nucleus. Okay, the DNA, somehow will send out all of the instructions out to make protein, the milk protein, to the ribosomes that are found, on the rough ER. So the protein is going to be made here, primary structure, secondary and tertiary. All of that packaging is going to happen in the Golgi apparatus. And then we'll release the milk protein in the form of a vesicle. And that vesicle is going to be released out through the body, through the cell, through exocytosis. But there's more to milk, more than protein. So we also have lipid, fat in the milk. That fat or lipid is going to be made by the smooth ER. There might be water. Water is just found in the cell, so that'll be released. And you can see how all of these organelles work together. And you'll find that the mitochondria is going to provide the energy for all of this to happen. The next organelle to talk about is the mitochondria. And the mitochondria is made out of two layers. There's an outer membrane, and an inner mitochondrial membrane. The outer membrane is smooth. The inner membrane has all of these ridges, or kind of like shelves on there. The mitochondria shelves are known as the mitochondrial crista, or the mitochondrial cristae. And this is where, it's known as the powerhouse of the cell. This is where most of the ATP molecules or the energy molecules are being made. Lysosomes and peroxisomes. Now these are different vesicles. So they are membranous sacs. In the case of lysosomes, they have enzymes that'll digest proteins, carbs, nucleic acid, bacteria, debris, and worn out cell parts. So as some of these organelles, like the other organelles, as some of them are going to age and die. So the cell would use the enzymes found in these lysosomes to digest them and get rid of all of that debris and worn out cell parts. If there is a bacteria or a germ trying to invade the cell, again, the enzymes found in these lysosomes will digest and get rid of that bacteria. So we want to think of the lysosomes as kind of like the garbage disposal of cells. The cells that you'd expect to have lots of lysosomes, for example, are your immune system. So white blood cells are huge on lysosomes. Another kind of membranous sac that also has enzymes, but a different set of enzymes, is known as a peroxisome. These enzymes can digest lipids, alcohol, and hydrogen peroxide. These peroxisomes are, for example, found a lot in the liver. So liver is responsible for detoxifying alcohol, detoxifying us from hydrogen peroxide. So anybody with liver problems would have a defect in the ability, or would in the ability to detoxify alcohol and hydrogen peroxide. For the microfilaments, microtubules, and intermediate filaments, well, these are all made out of protein. Hey, we kind of talked a little bit about them, so some of them are going to make that cytoskeleton that we talked about. And you can see here how all of these rods are kind of keeping the cell intact, keeping it open so it doesn't collapse, and making sure that the organelles are in their positions. And these microfilaments, for example, actin, okay, that's a protein that is very important for muscle contraction. We have intermediate filaments, and those are your cytoskeletal structures that we see here. They support also the nuclear envelope, which is right here. Microtubules, those are made out of a protein known as tubulin, and these make up your cilia, flagella, and centrioles. So if you're not sure what those are, we'll talk about them, I think, in the next slide or two. So your centrosome, or each centrosome is made out of 2 centrioles, and those are very close to the nucleus, and they really don't have a function until the cell is ready to divide. And then you'll see that each centriole will go to one side of the body, each centrosome will go to one side of the body and help in the process of dividing the chromosomes, so help in the process of mitosis. Cilia, some cells have cilia, which are like these eyelashes on the border of the cell, okay, and inside of each of this cilia, you would have a little bit of protein. Okay, so cilia, there are multiple cilia on the cell. If the cell is ciliated, again, there's protein inside of them. And the function of cilia is to move. It does like this wavy kind of movement. So, for example, the respiratory tract has cilia, and you can see here these power strokes, so that movement where the cilia, the respiratory tract, will be trying to kick out any foreign particles trying to get into the lungs. We also have cilia inside of the in the ovarian tube or the fallopian tube, and that helps to propel the egg towards the uterus if it is fertilized. Flagella is an extension of the cell membrane, and it is only one extension, okay, as opposed to cilia, there were multiple. Flagella is just one. The flagellum is responsible, it's like a tail that is responsible for the movement of cell, and the cell that has a flagellum is a sperm cell. So a little bit about the diseases of the organelles. Okay, so MELAS, that is a mutation in the genes found in the DNA of the mitochondria. So I know we talked about how DNA is usually in the nucleus, but there is a little part of DNA that is in the mitochondria. A mutation in that mitochondrial DNA will prevent the body from making, you have now mutated the powerhouse of the cell. So you'll see here that the person will not be able to make enough energy. Graves' disease is the inability to produce one of the lysosomal enzymes. So the person will not be able to produce myelin for nerve cells, and that leads to severe neurological damage. ADL, or adrenoleukodystrophy, is the lack of protein found in the membrane of the peroxisomes, and that leads to a buildup of fatty acids surrounding the myelin sheath, destroying it, and again leading to neurological problems. Now the cell, the nucleus, we haven't talked about that yet. We have the nucleolus in the middle. Okay, and the nucleolus is a very condensed part of it's made out of RNA and protein, and this is where ribosomes are made. You also have all of the chromatin, okay, that's made out of the DNA. This is where all the genetic material is. It has all of the manuals for protein synthesis. And then surrounding the nucleus is a double-layered nuclear envelope. You can see here these little pores in the envelope, providing some kind of passage between the nucleus and the cytoplasm. Next, we'll talk about movement in and out of the cell. So keep in mind that some movements are passive while others are active. Passive movements mean that they do not need ATP. They happen without any energy required. Active processes do require ATP. So for the passive ones, there's diffusion, osmosis, facilitated diffusion, and filtration, while the active processes are active transport, endo, exo, and transcytosis. For diffusion, you can see here in this experiment where if you put a cube of sugar, the sugar molecules are going to passively diffuse or passively move throughout the whole solution. And then if you leave it over time, you'll find that this is now, that sugar cube is totally dissolved and the water is very sugary. Okay, so again, this is a passive process, no ATP is needed, and where the molecules go from an area of high concentration and diffuse towards an area of low concentration. Diffusion can also happen even if there's a membrane in between. That membrane, though, has to allow the passage of both the salt and water in order for diffusion to occur. So you'll see that diffusion will keep on occurring back and forth, where both the salt or the sugar and water are going to keep on passing, back and forth until you reach a state of equilibrium. Facilitated diffusion is also another form of passive diffusion. It happens from an area of high concentration to an area of low concentration. Sometimes, though, as we mentioned before, that the cell membrane will not allow passage of water-soluble molecules. For example, sodium chloride, glucose, amino acids, those cannot go through the cell membrane. So they are going to go through these tunnels or through these channels, and that's what facilitated diffusion means. Osmosis is the passage of water across a semi-permeable or selectively permeable membrane. This membrane only allows for the passage of water. It does not allow for the passage of salt or sugar. So you'll find that water will pass from an area of high concentration of water to an area of lower concentration of water. An area of high concentration of water means that it is a diluted solution. An area of low concentration of water means that it is a concentrated solution. And this is important for the survival of red blood cells. So the osmotic pressure is the ability of a solution to pull water towards it. Okay, the more salt or the more protein or the more whatever it is that you're dissolving in water, the more concentrated a solution is, the stronger it is, or the better the ability it has to suck in water. So you'll see here that this is important again for plasma, for example, and the survival of red blood cells. Okay, so the osmotic pressure of the body is known as isotonic. So isotonic means that a solution has the same osmotic pressure as that of our plasma. If you put a red blood cell in an isotonic solution, then there is no net gain or loss of water to the red blood cell, and it remains healthy. But if you put the red blood cell in a hypertonic solution, that hypertonic solution is going to pull all the water out of the red blood cell, and you can see here that this red blood cell is now dehydrated. While if you put a red blood cell in hypotonic solution, okay, the RBC is going to absorb and gain lots of water, and you can see here that it keeps on growing until it bursts. Filtration is another kind of passive movement where things are filtered out. So you do have that filter, for example, the capillary wall. Capillary wall has very small pores that will allow for the very small molecules to be filtered out. Okay, so as the blood flows through the capillaries, because of that blood flow, because of the pressure of flow of water, so the hydrostatic pressure, that hydrostatic pressure is going to push a little bit of fluid and a little bit of the smaller molecules, while the bigger molecules are going to remain inside of the capillary. Now for the active processes, and these are, like I said, these need ATP. These go against the concentration gradient, from low to high concentration. And in an active transport, you have a carrier, okay? And that carrier is going to carry things in or out of the cell, again, against the concentration gradient. So we do need ATP in order for that to happen. So for example, sugar, amino acids, calcium, hydrogen, and the sodium-potassium pump. Sodium-potassium pump is a very famous pump that you want to be familiar with. Endocytosis would be the movement of a substance inside of the cell creating a vesicle. Sometimes this particle or the substance is just too large for any channels, too big to go through the cell membrane. So what the cell does is actually, engulfs it and creates a vesicle out of it, and that's how it kind of, takes it in. There are 3 different kinds of endocytosis depending on this particle. So if the particle is a liquid, so if the cell is drinking, that is known as penocytosis. If this particle is a solid particle, so maybe a bacteria or a virus, that would be engulfment known as phagocytosis. If the cell needs a receptor in order to attach to that particle, that is known as receptor-mediated endocytosis. And you can see here that is the cell membrane with the receptors on top, and these are going to attach to that receptor first and then become engulfed. So this is an example of receptor-mediated endocytosis. Exocytosis is the exact opposite of endocytosis. So exocytosis means that a vesicle is going to be released out. So this vesicle is going to attach to the cell membrane and then open up and release whatever was in that vesicle. For example, the release of neurotransmitters from nerve cells. Transcytosis means that things go in on one side of the cell and then exit on the other. So to go through the cell or trans the cell. So again, it involves endocytosis on one end and then exocytosis on the other end. And this is how HIV can cross the lining of the anal or the vaginal lining, because it is able to hide itself through the mucosal lining of the anus or the vagina by going in one end and exiting through the other. Next on our list is the cell cycle, which talks about the life cycle of the cell. Okay, so there are different stages. We first off have the interphase, and then there is mitosis, and there's also cytokinesis. So the interphase is the part of the cell that you can see here that is kind of makes up the majority of the cycle, and that's when the cell is actually performing its function. It also might be preparing for cell division. Mitosis is where the nucleus is dividing, and cytokinesis is where the cytoplasm is dividing. So let's take a look at these different phases of interphase. So interphase, again, is the part where the cell is pretty much performing its function. Right after division, you have a stage of growth. That's the G1 phase. So the cell is going to make more organelles. It is going to start performing its function. And then it reaches this restriction point where it has to decide, am I too old of a cell? Am I unable to perform my function? So I'm going to perform what is known as apoptosis, where the cell pretty much just destroys itself. Or am I good enough and I am going to remain specialized and keep on doing my function? Or am I a cell that needs to divide? So it will proceed to the division. In that case, it has to go through S phase, and S phase means that I'm going to duplicate or synthesize more genetic material. So the DNA In order for me to split this DNA among two cells, I have to make another copy, and that occurs during that S phase. G2 phase is another phase where I need to duplicate all of my other organelles. And then we can move on to mitosis. Okay. So here again, you can see the different phases on here, the interphase and S, G1, and G2 phases. Then we move on to mitosis, as I mentioned. Mitosis, that is the division of the nucleus, while cytokinesis is the division of the cytoplasm. So the phases of mitosis are prophase, metaphase, anaphase, and telophase. You can remember them as past my anatomy test. Okay, and I'm going to use the picture to kind of illustrate these changes. So in prophase, what happens is that the chromatin that is very, kind of entangled inside of the nucleus is going to condense to make these X-shaped chromosomes. And then you see here that the nuclear envelope starts to disappear. So this is what happens during prophase. In metaphase, you'll find the chromosomes lining up in the middle, and then you can see these mitotic spindles. They kind of look like spider webs that are coming out of the centrioles. These mitotic spindles are going to attach to the middle of the chromosomes, known as the centromere. Okay, and you see that all of the chromosomes are now lined up in the middle of the cell. That again is your metaphase. In anaphase, we are going to pull the chromosomes apart from each other. So you'll see that one chromatid will go to one pole of the cell. In telophase, now these chromosomes are going to collect together again, and a new nuclear envelope will be developed. Towards the end of telophase, cytokinesis starts, and that is the division of the cytoplasm, where you see there is this contractile ring right in the middle, and this cleavage keeps on getting deeper and deeper until we separate into two cells. Here you can see the cytoplasmic division, where again that cleavage happens in the middle, and then it keeps on progressing until the cell divides into two daughter cells. So how does the cell control all of this division? Some cells are going to keep on continuously dividing. For example, skin cells, intestinal cells, blood cells. These are cells that are continuously being destroyed and we have to replenish, while other cells do not divide as much. For example, neurons, some of them don't even divide at all. And each cell has a timetable, so it knows how many times it can divide, and it uses the chromosome tips known as telomeres. Each time the cell divides, a part of this telomere is lost. So when the telomere shortens, that's when the cell knows that it is too old of a cell and it needs to go through apoptosis. Sometimes we can control cell division by certain hormones and growth factors. Contact inhibition, for example, is another thing. So healthy cells, for example, healthy skin that is intact without any wounds on there is not going to divide. So there is an inhibition of cell division due to contact. But when somebody gets hurt, if there is a wound, there is a disruption of that contact, now these cells know that they have to rapidly divide in order to heal that wound. So a little bit about tumors, since tumors are really abnormal growths. We've got two different kinds of tumors. There are benign tumors, and those are the ones that remain locally. They do not spread to the rest of the body. And then there are malignant tumors, also known as cancerous tumors. These are invasive. They can invade the blood vessels, the lymphatics, or even metastasize and spread to the rest of the body. We have two different kinds of genes that might cause cancer, or an imbalance of them could cause cancer. So we have oncogenes and tumor suppressor genes. Oncogenes are tumor-making genes. Okay, if these genes are overexpressed, these can lead to cancer. And we also have tumor suppressor genes. Okay, so these tumor suppressor genes are obviously trying to prevent cancer from happening, but sometimes if they are inactivated or if they are removed, we've lost that tumor suppression, and that would also might lead to cancerous changes. Here you can see a little bit of the steps of the development of cancer. So you can see here that, there's a imbalance between the oncogenes and the tumor suppressor genes. Either the oncogenes are turned on or the suppressor genes are turned off. So now we have lost the control of the mitosis of the cell. You can see here that, we have too many cells being made. These are usually abnormal cells. They're not healthy. They do not look like the mother cell. They start invading locally, the local tissue surrounding it. They start invading the blood vessels. And you can see here that through the venous system, they are going to start, it might start invading, reaching other organs as well. Now back to normal cells, starting from the embryo where we had only the sperm and the egg fertilized to make that zygote, that is a stem cell. Okay, so a stem cell has the ability to become any cell in the body. So that stem cell is going to keep on dividing, making more and more copies of itself, and then it has to differentiate. So the differentiation process is the specialization of cells, where a stem cell becomes maybe a nerve, it could become a muscle, maybe intestine. So that process of specializing is known as differentiation. A stem cell has the ability to make other stem cells, or it can also start making what is known as a progenitor cell. A progenitor cell is a partially specialized stem cell, okay, meaning that it is kind of like that bridge between a stem cell and an actual well-differentiated cell. So a progenitor cell has the ability to become anything within one organ system. So for example, if a progenitor cell commits to becoming part of the nervous system, it has the ability to become either a brain cell or a nerve cell, or maybe a Schwann cell, which are different cells in the nervous system, but it cannot become, say, a red blood cell. Okay, it has committed to that one system, which gives these different cells different strengths. So we have a totipotent cell, which is pretty much, has the total potency, has a total strength to become anything. For example, fertilized eggs. Okay, these are cells that can become any kind of cell in the body. Pluripotent means that it still has some kind of potency to become plural or different kinds of cells. But again, it's a limited, it has already committed to a certain system. For example, these progenitor cells are definitely pluripotent cells. We can see here the process of differentiation. So you see here the very first step would be the fertilization of the egg. So the sperm fertilized the egg. We have now a fertilized egg, so this is definitely a stem cell. That stem cell has the ability to make more stem cells, or it can also start making a progenitor cell. Okay, we'll see here that these progenitor cells, for example, in this instant, dedicated themselves to make nervous tissue, while this set of progenitor cells dedicated itself to make, say, parts of the integumentary system. Okay. So this stem cell is a totipotent cell, while these progenitor cells are pluripotent. So how can we use stem cells to study and treat diseases? Okay, we need stem cells and progenitor cells for growth and healing. Okay, we are continuously making, or in the need of making, fresh cells throughout our life. Regenerative medicine is a field of study that uses the potential of the body to make these new cells to treat diseases and injuries, and this includes what is known as stem cell technology. Sources of stem cells for research could be donor stem cells, like cells from the umbilical cord, or stem cells from the patient themselves, for example, from bone marrow. So bone marrow is full of stem cells that we could use, or sometimes we can even take cells that have grown into into the mature form, and we can reprogram them to go back to a stem cell. Next, for cell death, there are different kinds. There's apoptosis and necrosis. Apoptosis is programmed cell death. And we talked about that in the cell cycle, where the cell reaches that restriction point and decides what it has to do. So if it decides that the cell is too old, it will, through a program, kind of go through these steps of self-destruction. For example, if you get sunburned, that peeling off of the skin, that is apoptosis. Necrosis, though, is cell death from damage. Okay, so burns, for example, is an example, or myocardial infarction, which would be a heart attack. That is necrotic cell death. That is not normal process. That is a death that happened due to damage.